presenter, which is Dr. Michelle Duenas. I'm sorry, Michelle. I never asked you how to pronounce your last name.
Oh, that's okay. It's Duenas. Can you hear me?
Duenas, yes, from St. Vincent College.
Do you see a picture of some bees right now?
Yes. Yep.
Okay. And now, let's see if this works. And now, do you see my slides without the notes?
Yes. Yeah.
Okay, great. Thanks everyone for being here for my talk today. I'm enjoying learning so much about other groups' research. And the main goal of my talk today is to look for collaborators. But I wanted to talk about a pilot project that I launched this year with Dr. Al McDonald at Chatham. And so to talk about this, I'm going to talk a little bit about how I got into Spotted Lanternfly recently, how we recruited people for the citizen science project, what we plan to do for the future, and then ask for calls for collaborations and any ideas any of you might have. So here at St. Vincent College, we're a small liberal arts college, and so I don't advise master's or PhD students, but I do advise 3-semester senior research projects. So they're kind of like about a chapter of a master's thesis, and Claire was one of my students that graduated last year, and she was interested in studying insects and evolution. She's now in graduate school at the University of Iowa studying insects and evolution. I don't know if she's studying spotted lanternfly still, but I suggested we study these new emerging pests that would probably be pretty easy to find and do genetic analyses on, and so that's what we worked on. But Claire did not have a car, and because of my heavy teaching load, I didn't have tons of time to collect samples, and so we recruited from the St. Vincent community, and some of our friends and my roller derby team helped us collect samples, and so we recruited people from the community to help us collect samples. And that proved very fruitful. We got over 114 samples for Claire's project. And one of the methods that we used for looking at her spotted lanternflies is we wanted to test out wing morphometric methods and see if we could see any differences in the samples she collected based on wing venation. And so we used the landmarks that were from this study here. And so Claire mounted all of these wings on slides and then used the software program MorphoJ to map them and then do principal components analysis of the variation in wing shape. Probably unsurprising to everyone here, we had such a small sample size from a very small region, and they're so recent. Here in Pennsylvania, there was not any sort of distinct pattern in their wing shape at all. Claire also used a really small, because this is a undergraduate research project, we only used 4 microsatellite loci. But Claire also looked at 4 microsatellite loci from this Kim et al. Paper, and probably also unsurprising to everyone, there was complete gene flow between all of our samples. So if you don't know how to read this, this is a structure graph where we looked at the genetic diversity in the samples, then it tries to sort samples into populations. And so for this analysis, we were testing 2 populations, and every single sample was assigned to about 50% population 1 and population 2, which means they have complete gene flow with each other. So not much from this senior research project, but because there's a lot of interest locally, I decided that because of what we were able to accomplish and because of the interest we had in Claire's project, with a collaborator who I met because we both play roller derby in Pittsburgh. Dr. McDonald is a biochemistry professor at Chatham, also another small liberal arts college. We teamed up to try to launch a larger citizen science project here in Pittsburgh to see if we could get interest in people collecting samples that we could use for future DNA or RNA work or for biochemical analyses. And this is the logo that I designed to make the project look a little more official. And so the way that we recruited people, well, this was more accidental, I guess, but a lot of people who are here today were featured in this National Geographic article. And when Jason re-released it in August, he mentioned this in there, and then that was picked up by the Tribune, which is a Westmoreland County paper. They featured the research in there. We also posted it on my lab Instagram page. And then once it got picked up by the local news, then Dr. McDonald and I went on a local talk show to recruit participants. We were featured on a couple local news spots. And once that happened, it kind of blew up. I expected maybe 50 people to sign up to participate in this, but in the end, we ended up with 425 people signed up. And so what they did, if they were interested, is they went to my lab website and they filled out this participant application form. And then they put in their address, and then we mailed them a kit. And this is what the kit consisted of. So because I was prepared, I had about 50 sample kits prepped to send to about 50 people, but then 425 people signed up, and I really wanted to encourage this participation. So undergraduates in a bunch of classes at St. Vincent got extra credit or work study hours for making kits for us. And in the kit, we put a 50 milliliter Falcon tube in a separate bag, along with instructions for collecting the samples. And then I created basically like an online ship account, and we mailed out all of these to each of the individual participants. And then in the kit were instructions. So what we had, because my background is in evolutionary ecologist, my main study area is bees. But I'm particularly interested in genetics. And so we had people, we sent them the kits without alcohol and asked them to add rubbing alcohol to the samples. And then we also asked them to collect a minimum of 5 female and 5 male samples. And so we also included a guide of how to identify the males from the females. And then this was the data collection sheet that we included with each of the kits. Just so you can see a little bit of information about the people who signed up to participate in this. Because Dr. McDonald and I are really interested in teaching, because we're at small liberal arts colleges, we also really want to take this further and maybe develop K through 12 lesson plans. And so we were really interested in teachers signing up. But most of the people who signed up were individuals that were just collecting at their place of residence. And most of this other category is people who wanted to get 2 kits so they could collect at their house and also their place of business. And most of the participants, we limited it to Allegheny and Westmoreland County just for sheer logistics and being able to send out enough kits. But most participants were from Allegheny County because they aren't super present in Westmoreland County yet. And then what we're doing now, so people collected these in the fall. The kits did go out a little late because I was not expecting that many people to sign up. But then we are now having people drop off their samples to us. I was out of the country in Mexico at the end of the year studying bees, but now I'm back, and so I have time to take and process all these samples with my students. And so one of the weekend drop-off locations is a coffee shop that's in the Carnegie Library in Pittsburgh, because I am a weekend barista there. And so I've gotten to meet a lot of participants in person and make them coffee if they drop off their samples. The other drop-off location is where I'm located right now, which is the Science Pavilion at St. Vincent. And if the building is closed, we have a bucket outside full of rocks because our original bucket blew away in some heavy winds, but people can drop their samples off in there. We've only had sample drop-off locations released for a week and a half, and we've gotten back 3 so far. And because every participant put their email address and contact information, we'll be following up and making sure that people turn in their samples and their kits. I did think we've got a lot of children participating. One participant came dressed up to drop off her samples just this week. And then there was a school district superintendent in Sewickley that bought her own kits for every second grade class in her school district. So we're going to have a lot of samples from them. And so she had the students fill out the data sheets, and I thought people would enjoy some of the data that she got from her grade school students. And so that's where we are right now with the project. It's still very, very early. We kind of did this first year as a pilot study to see what we were capable of. And then we're still hoping to get all of those samples back so I can report on that later. But what are we planning to do with the future? So we want to sample the same locations every year. We want this to be a long-term study. And so we're hoping these participants that participated this year will continue to participate and collect samples for us every year. Um, Depending on funding situations, we might expand out to Butler and Washington counties if we can get enough money to make all of these kits. Um, what we'd also like to do this year is select some of the participants and their homes as places for liquid nitrogen collection and storage of samples that then we can maybe use those samples for extracting RNA, looking at secondary metabolites, or doing further studies where liquid nitrogen frozen and ultra cold frozen samples are useful. What Dr. McDonald and I plan on doing is having our my ecology, evolution, and invertebrate zoology classes in the future are going to use these samples to practice doing DNA extraction and analyzing genetic data. And so some of the data I plan to collect with my students is just basic genetic data from the flies themselves. But depending on what they're interested in, they could do metagenomic analysis of their gut contents or really anything they can think of, any sort of metagenomic data they might want to collect from these samples, any sort of genetic data. And then Dr. McDonald is going to have their biochemistry class process samples and do biochemical analyses on the liquid nitrogen preserved samples. And again, both of us are using these as samples for designing independent student research projects. So it'll be up to the students what kind of data they want to analyze. We also want to partner with K through 12 schools and teachers to create lesson plans around spotted lanternflies. But what we would really like, and what my kind of like grand aspiration for this project is to become a resource for other spotted lanternfly researchers. We have a lot of facilities here at St. Vincent, but we are a small liberal arts college focused on teaching, and so we don't have a ton of funding for research. But I would love to become a sample bank for researchers doing lots of different kinds of studies. So what we're already doing with the samples is preserving them for genetic analysis. We're also preserving them for RNA and for metabolomic analysis. And all of those samples we would love to share with other researchers who have more funding. Here are just some ideas I even added to while I was listening to the talks today, but our citizen science participants are very eager to collect and help out researchers. And so any other kind of data that you think citizen scientists could collect for your project to help you, we would love to recruit them to do that. So any host plant data, testing any traps, any behavioral observations, or anything else you think citizens could collect for you, we would love to put our group to work for you to help you and become a resource to researchers anywhere, really. And so with that, I want to thank all the people I've already mentioned throughout this talk and all of you for listening. And here's a QR code to link to where we have all of our information about the archive we're building and my contact information if anybody would love to take advantage of our samples. And I'll take any questions.
All right. Thanks, Michelle. That was great. Does anybody have any questions for Michelle?
Oh, I should, I'm actually going to stop sharing so I can see if there's anything in the chat.